Welcome to EQBloom, where science meets self-awareness and biology meets the future. Today's topic might sound like science fiction, but it's very real and very now. CRISPR and the future of gene editing. What if I told you that we now have the power to edit life itself, to change genetic instructions that define who we are? This isn't just a tool, it's a revolution. It's poised to change everything from how we treat disease to how we define humanity. So how exactly does it work? Where did it come from? What does it promise? And what dangers lie ahead? Let's break it down. CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeats. Bit of a mouthful, right? But it's a crucial part of the immune system in bacteria. Yes, CRISPR was not invented in a lab. It was discovered in bacteria. Think of it like this. Bacteria get infected by viruses too, just like we do. To defend themselves, they evolved a memory system, like a genetic scrapbook, of past viral invaders. These stored fragments help them recognize and destroy future attacks. CRISPR is this defense system. When a virus infects a bacterium, it stores a bit of that viral DNA in its own genome, right in a CRISPR sequence. When the same virus comes back, the bacteria produce an RNA guide from that sequence, and that guide helps a special enzyme, Cas9, find and destroy that viral DNA. Now here's a leap. Scientists figured out that they could hijack the system. By designing custom guide RNAs, we can direct Cas9 to cut any DNA sequence we want. It's fast, it's accurate, and it's cheap. In 2012, researchers Jennifer and Emmanuel published the first paper showing that this bacterial system could be used to edit genes in other organisms. 8 years later, they won the Nobel Prize in Chemistry. And CRISPR hasn't slowed down since. So let's dive into the mechanism. CRISPR works like a genetic GPS and scissors combined. Here's a step-by-step breakdown. Guide RNA synthesized to match the DNA sequence we want to target. This RNA binds to Cas9, a protein that functions like molecular scissors. The Cas9 guide RNA complex patrols the genome, scanning for a perfect match. When it finds it, snip, Cas9 cuts both strands of the DNA. Now here's where the magic and challenge begins. Cells don't like broken DNA. They immediately try to repair the cut using one of two pathways. The first one is non-homologous end joining, NHEJ. This is fast but messy. It often introduces small errors, deletions, or insertions that can disable a gene. This is useful when we want to knock out a faulty gene. The second one is homology-directed repair, HDR. This pathway is more precise. Scientists can introduce a custom DNA template along with the cut, and the cell may use that to patch the break, effectively replacing the original sequence with an edited version. In short, CRISPR is like a highly targeted find and replace tool for your genome, and this ability opens the door to extraordinary possibilities. Let's look at what CRISPR is also doing in the real world. Medicine. In 2020, a landmark clinical trial used CRISPR to treat patients with sickle cell anemia, a painful and life-shortening blood disorder. By editing stem cells, researchers effectively cured the disease in early patients. Other trials are exploring CRISPR for leukemia, beta thalassemia, and even blindness. Number 2, agriculture. CRISPR-edited crops are being engineered for drought resistance, higher nutritional value, and pest resistance, without the need for foreign DNA, which helps avoid the stigma of GMOs. Number 3, research. Scientists use CRISPR to create custom animal models of the disease, such as mice with humanized immune systems or Alzheimer's-related mutations, speeding up drug discovery and understanding. Number 4, public health. Through a technology called gene drives, CRISPR could spread genetic changes through wild populations. For example, to eliminate malaria-carrying mosquitoes by making them sterile or resistant to the parasite. It's not just a theory, it's action, but the deeper we edit, the more complex the consequences. Of course, this power doesn't come without profound ethical dilemmas. In 2018, the world was stunned when Chinese researcher He Jiankui announced he had edited the genomes of twin embryos to make them resistant to HIV. These edits were made before birth and passed on to future generations, a process known as germline editing. The backlash was immediate. Why? Because germline editing isn't just about one person, it affects their children and their children's children. It's permanent and mistakes could be disastrous. Then there's the issue of equity. Who gets access to gene editing? If only the wealthy can afford it, could it deepen social divides, creating a genetic upper class? And what happens when people start editing for non-medical reasons? Height, intelligence, even eye color. The specter of designer babies raises fears of eugenics and perfectionism. Science moves faster than legislation, so we need robust global ethical frameworks to guide how CRISPR is used before the genie escapes the bottle entirely. CRISPR is just the beginning. Next-gen tools are already here. Base editing swaps a single letter of DNA without cutting both strands, like fixing a typo with a pencil instead of scissors. Prime editing, a more refined approach that can insert new sequences with fewer errors. The future includes personalized medicine tailored to your unique genetic profile, 3D printed organs, seeded with edited stem cells, possibly even life extension, cancer vaccines, or neurological repair for disorders like ALS or Parkinson's. It's a frontier filled with promise and peril. We may soon be able to not just treat disease, but to prevent or improve the limits of the human body. But as we approach the ability to rewrite life's blueprint, the question becomes less can we and more should we. We are the first species in Earth's history to read and edit its own code. With CRISPR, we've unlocked a new language, one written in the A, T, C, and G of DNA. But power without wisdom is dangerous. As we continue editing the story of life, we must remember that every cut, every insertion, every fix echoes through time. So, what would you change if you could rewrite your genetic code? Would you remove a disease, enhance your memory, extend your life? Or would you pause and ask what it means to be human? Subscribe to EQBloom for more science that shapes your future and your biology, because understanding the tools of tomorrow is how we take responsibility for today. Thanks for watching.